So I want you to think about the last time you had a routine blood test. You get that little pinch in your arm, you go about your week, and then maybe a few days later, a notification pops up on your phone.
Right, usually from a patient portal or a health app.
Exactly. So you open the app and you just scroll past this huge wall of medical acronyms, right?
Mm-hmm.
You see that ALT is in the normal range, you breathe a sigh of relief, and you just move on with your day.
Yeah, and we've really been conditioned to treat those results as this, simple binary. Like if the text is green, you're safe. But if the text is red, you panic.
Right, we want our health data categorized neatly.
We do, but a single number on a screen actually obscures this massive, highly precarious chain of biological and physical events.
And that is exactly what our deep dive is about today. We are pulling back the curtain on that exact chain of events by looking at a stack of biomedical pathology notes from the LabPathPro Audio Academy.
Which is such a great resource, by the way.
Oh, it's fantastic. So we're decoding the hidden world of alanine aminotransferase, which most people just know as ALT.
If they know it at all, right?
Right, but this isn't just a quick look at, what this enzyme does in your body. We're actually following the complete journey of a patient's sample, from the second the needle pierces the vein all the way to the final authorized result.
Because understanding that workflow,
Right.
It's fundamental to grasping why we can trust modern medical diagnostics in the first place.
Yeah, we casually throw around terms like liver enzyme.
Right.
But the actual biochemical role ALT plays, paired with the intensely complex physics and, the quality control required to measure it, it really represents a peak achievement in laboratory medicine.
It absolutely does.
So let's start with the biology. Before we can even measure ALT in a high-tech lab, we really need to know what it's doing inside your body right this second.
Well, ALT stands for Alanine Aminotransferase, though older medical charts sometimes list it as SGPT. Yeah, it stands for serum glutamate pyruvate transaminase, but they refer to the exact same thing.
Good to know if you're looking at an old file.
Exactly. And its primary characteristic is that it's an intracellular enzyme, meaning it lives and works primarily inside your cells.
So not floating around in the blood normally?
Right, mostly inside the hepatocytes, which are the main functional cells of the liver. You'll find, trace amounts in the kidneys and skeletal muscles, but the liver really holds the lion's share.
Okay, so what is it actually doing in there?
Inside those hepatocytes, ALT is basically a workhorse for amino acid and nitrogen metabolism.
The chemistry here from the notes is highly specific, right? It catalyzes the transfer of amino groups.
Yes, it takes two substances, alanine and 2-oxyglutarate, and it facilitates a reaction that converts them into pyruvate and glutamate.
Which I assume is pretty important.
Oh, it's a fundamental metabolic process for energy production and managing nitrogen waste. But, the critical detail for diagnostics is the boundary between where that metabolism happens and where we actually draw the blood.
Right, because under normal healthy conditions, the vast majority of ALT stays locked safely inside the liver cells doing its job.
Precisely. Only a really microscopic fraction naturally turns over and ends up in your circulating blood.
So the way I was picturing this from the notes, it's like, I picture the liver as a highly secure, specialized chemical factory.
Oh, I like that.
Yeah, and the ALT enzymes are the factory workers. So on a normal day, they clock in, they stay inside the building, they run the metabolic machinery, and only a tiny handful of them wander outside the gates.
Right.
But if the factory sustains structural damage, like if the walls literally break open, those workers flood out into the surrounding streets, which in our bodies is the bloodstream.
Taking that factory analogy a step further, the concentration gradient is really what makes this so useful for clinicians.
How so?
Well, because the liver holds such a massively high concentration of ALT compared to other organs, a sudden spike of these enzymes in the blood is a blazing, highly specific indicator of hepatocyte injury.
So it points the finger directly at the liver.
Exactly. It makes it a much more specific marker than its frequent partner enzyme, AST.
That's aspartate aminotransferase, right?
Right, which is found in much higher concentrations in places like the heart and skeletal muscle. So AST is less specific to just the liver.
I imagine that specificity is exactly why a clinician actively hunts for ALT when they order a blood panel. They want that direct evidence of factory damage.
They do. An ALT test is a frontline tool. They use it to assess conditions like viral hepatitis, fatty liver disease, alcohol-related liver disease, and drug-induced liver injury.
And they track it over time too, right?
Yes. longitudinally to monitor disease progression or to see if a patient is responding to a therapy over months or years.
Okay, there's a massive misconception here that we really need to clear up because I hear this all the time. People constantly refer to ALT as a liver function test.
Ugh, yes, it is an incredibly common misnomer, even among some healthcare professionals, honestly. ALT does not measure function.
It measures injury.
Exactly, it measures injury.
Meaning, you could have a damaged liver that is leaking ALT everywhere, but the liver is still technically keeping up with its functional duties for the time being, like producing proteins or clearing toxins.
Yes, and conversely, an elevated ALT doesn't automatically mean you have some devastating infection like hepatitis.
Right, because numerous things can cause that damage.
Right, anything from taking way too much paracetamol to metabolic issues, they can damage those cells and elevate the enzyme. This is why ALT is rarely interpreted in a vacuum. It's part of an ensemble cast.
Let's define that ensemble cast, actually, because they all show up on that same app notification we talked about.
Sure.
You've got AST, which we mentioned, ALP, which is alkaline phosphatase, often pointing to bile duct or bone issues.
Right.
GGT, or gamma-glutamyl-transferase, which is highly sensitive to alcohol and biliary tract disease, plus bilirubin and albumin.
And the clinician reviews that entire panel alongside of the patient's clinical history. I mean, you just cannot diagnose a patient based on a single metric.
Well, let me push back on that from the perspective of a patient just looking at their phone.
Okay, go for it.
If my ALT is completely normal, the text is green. Doesn't that mean my liver is 100% perfectly healthy and I have a free pass to eat or drink whatever I want?
I would strongly advise against treating a normal ALT as a clean bill of liver health.
Really?
A normal result absolutely does not exclude liver disease.
Is that because the damage might not be actively leaking enzymes at that exact specific moment?
Precisely. There are forms of chronic liver disease, including advanced cirrhosis, where the ALT levels can fluctuate or even appear entirely normal.
Wow.
Yeah, the cellular destruction might be a slow, smoldering burn rather than this acute explosion. Or the liver might have already sustained so much long-term scarring that there just aren't enough healthy cells left to leak significant amounts of ALT.
Oh, that makes sense. The active leakage isn't the main issue anymore.
Exactly. That is why your doctor evaluates your symptoms, your history, your lifestyle, and that entire ensemble cast of markers.
Okay, so the clinician evaluates the history and decides to order the panel. The needle goes in, the blood goes into the vial. Now, getting that blood from your arm to the laboratory analyzer is called the pre-analytical phase.
Yes, it is.
And looking at the notes, this seems to be the most dangerous part of the entire process.
It is arguably the most perilous part of the whole journey. The workflow moves from patient identification to blood collection, selecting the correct chemical tube, transport, laboratory reception, and finally sample preparation.
That's a lot of steps.
It is. And the vast majority of laboratory errors occur right here, long before the sample ever touches a machine.
It makes sense. It's kind of like trying to measure the exact acidity of a really delicate soup, but the delivery driver accidentally spilled a bottle of vinegar into the container on the drive over.
That's a very apt way to look at it.
Right, because the analytical tool at the lab might be perfectly calibrated, but the sample itself has been fundamentally altered.
The physical and chemical handling of the sample dictates the reality of the result.
Like the tubes they use.
Right, the phlebotomist must draw the blood into a very specific environment. Depending on the analyzer the lab uses, they might require a serum tube or maybe plasma tubes treated with lithium heparin, sodium heparin, or EDTA.
And if they use the wrong one?
If they use the wrong anticoagulant, the chemical additives in the tube can completely inhibit the enzymatic reactions the machine tries to run later on.
Oh, wow. And beyond just the chemistry of the tube, there is the physical handling, right? Like adequate mixing, proper centrifugation to separate the liquid serum from the heavy blood cells, and temperature-controlled transport.
Yes, which introduces a highly disruptive pre-analytical error known as hemolysis.
Yes, hemolysis. This was fascinating in the notes. Walk us through what actually happens to the blood cells here.
So hemolysis occurs when red blood cells physically burst open.
Just pop.
Exactly. And this could be triggered if the blood is drawn too forcefully with a syringe, or if the needle gauge is too small and it creates shear stress, or even if the tube is shaken violently instead of being gently inverted.
Or extreme temperature changes in transit, right?
Yes, that too. The membrane of the red blood cell literally ruptures.
And because red blood cells contain their own internal enzymes, Bursting them dumps all those contents directly into the serum we're trying to test.
Exactly. It falsely spikes the ALT levels in the sample. So the analyzer will read a high ALT, but the patient's liver is perfectly fine.
The cellular injury actually happened to the blood cells inside the plastic tube during the drive across town.
Right. It leads to an entirely inaccurate clinical picture, which is really why specimen quality is the foundation of all diagnostics.
Okay, so assuming the sample survives the journey, it arrives safely, no hemolysis, correct tube, it's perfectly centrifuged. How does the laboratory analyzer actually count these microscopic ALT enzymes?
Well, the surprising answer is that it doesn't count them at all.
Wait, it doesn't?
No, you cannot tally up enzymes like inventory in a warehouse. Modern analyzers, such as the Abbott Alinity systems, they measure enzyme activity, not mass or particle count.
Oh, okay. And the chemistry behind this is incredibly elegant. According to the notes, laboratories utilize an IFCC standardized enzymatic assay.
Yes.
The IFCC is the International Federation of Clinical Chemistry, meaning they set a universal recipe so a lab in New York and a lab in London are speaking the exact same chemical language.
The standardization is absolutely key, and the assay involves a highly controlled sequence.
Walk us through it.
First, there is pyridoxal phosphate activation. This acts as a necessary coenzyme to ensure all the ALT in the patient's sample is primed and ready to work.
Got it. Waking the workers up.
Right. Then the machine introduces what we call a coupled reaction, utilizing a secondary enzyme called lactate dehydrogenase.
So they're essentially forcing the ALT to do its normal day job, that amino acid metabolism we talked about earlier, but they are doing it inside the machine under a microscope.
Exactly. They provide the ALT with the specific chemical puzzle it evolved to solve. And as the ALT does its work, the machine monitors a secondary linked reaction, which is the oxidation of a coenzyme called NADH.
Nicotinamide adenine dinucleotide.
Yes, that's the one. The machine measures this process as a rate reaction using a photometer set to a very specific wavelength of ultraviolet light, 340 nanometers to be exact.
Okay, let me pause on the light measurement because the mechanics here are brilliant. The notes point out that the absorbance of the light actually decreases during the reaction.
It does.
Which is weird, right? Usually when we think of a chemical test, like, I don't know, a pool pH strip, a color gets darker or stronger the more of a substance is present. But here, the light absorbance is dropping.
Right, because we are measuring the consumption of NADH. See, NADH absorbs ultraviolet light very strongly at 340 nanometers. But as the ALT enzyme drives the reaction forward, it uses up the NADH, oxidizing it into a different form, NAD plus O, which does not absorb light at that wavelength.
So the test is essentially like an invisible windmill.
An invisible windmill.
Yeah, like you can't see the wind, which is the ALT, but you can measure exactly how fast it's blowing by watching how quickly the grain gets ground up and disappears.
That is a perfect analogy. Building on that, the liquid actually absorbs less light over time because the grain, the NADH, is vanishing.
Wow.
The analyzer isn't looking at the ALT at all. It is calculating how fast the ALT is chewing through the chemical substrate. So the faster the liquid loses its ability to absorb light, the more ALT activity is present in the patient's serum.
Just the physics and chemistry required to produce that single data point on an app is absolutely staggering.
It really is.
But if we are relying on an invisible reaction and a decreasing light signal, how do we know the machine isn't just, malfunctioning? What if the photometer bulb dims or the chemical reagents degrade?
Yeah, that brings us to the most critical layer of the laboratory, which is the quality assurance pipeline and highly trained biomedical scientists.
Right.
An analyzer does not just run patient samples blindly. The entire process is anchored by calibration, which establishes the mathematical baseline for the light absorbance. And following calibration is internal quality control, or IQC.
This is where they run fake samples with a known exact amount of ALT to see if the machine hits the target, right?
Yes, and labs apply strict statistical frameworks to this, known as Westgard rules.
Go Westgard rules.
Sounds intense.
They are. They rely on standard deviations from the mean. So if a quality control sample reads outside of these highly specific statistical boundaries, say, it's consistently reading a little too high over several days, it flags an error.
And then what?
The cardinal rule of any accredited laboratory is that a patient result must never be released if the quality control has failed.
Ever.
Ever. Furthermore, labs participate in external quality assessment, or EQA. An outside regulatory agency sends blind mystery samples to the lab.
Oh, like a pop quiz.
Basically. The lab tests them and sends the results back to ensure their machines are perfectly harmonized with other laboratories nationwide.
And after all the robotics, the light-absorbing algorithms, and the statistical rules, every single result still goes through verification by a biomedical scientist before it is authorized for the doctor to see.
Well, let me play devil's advocate here.
Sure.
We live in an era of unprecedented automation, right? We have algorithms that can literally drive cars. Why can't we just let the machines run the entire diagnostic show, cut out the middleman, test the blood, and automatically email the patient their results the exact second the machine finishes the math?
I mean, it is a tempting thought for efficiency, but automation is merely a tool. It is not a scientist, and it lacks clinical judgment.
Right.
A machine might flag a sample for a pre-analytical error like hemolysis, but it takes a biomedical scientist to evaluate the clinical context and recognize when a sample requires outright rejection.
Because releasing it would be dangerous.
Because the data would be dangerously misleading for the physician. A great example of this human oversight is the Delta check.
Okay, Delta checks, how do those work?
So this is when the laboratory information system compares your result today with your result from a week or a month ago. If your ALT was 30 last week and 3,000 today, the machine will just blindly output the 3,000. It doesn't care.
Yeah.
But a biomedical scientist will halt that result. They will investigate the discrepancy.
They have to figure out if it is a catastrophic, acute liver failure, or if, say, a nurse accidentally drew the blood from an IV line that was contaminated with saline, diluting or altering the sample.
Exactly. They might mandate a repeat analysis or institute manual dilution protocols if a patient's enzyme level is so incredibly high that it exhausts all the NADH in the machine instantly, breaking the readable range of the assay.
Wow.
They evaluate critical values and call the doctors directly. You just cannot code human clinical intuition and scientific skepticism into a photometer.
No, you definitely can't. So to really cement everything we've just unpacked, let's apply this massive journey to a real-world clinical scenario. Let's put the puzzle pieces together.
Okay, I will take the role of the clinician. Walk me through the case.
All right, we have a 45-year-old patient. Their body mass index, or BMI, is 34. Their primary complaint is generalized fatigue, just feeling constantly tired.
Okay. The clinician ordered the full ensemble cast of liver tests. The blood was drawn perfectly, no hemolysis. The Westgard rules passed. The biomedical scientist verified the data, and the results are as follows.
Weigh it on me.
The ALT is significantly raised. The AST is mildly raised. But the ALP is completely normal, and the bilirubin is normal.
Yeah.
How do you interpret this?
Okay, well, first, we categorize the pattern of injury by looking at the relationships between the enzymes.
Right.
The ALT and AST are elevated, but the ALP and bilirubin are normal. This specific divergence points directly to hepatocellular injury.
Meaning the liver factory itself, the hepatocytes, are taking damage.
Exactly. If the issue were in the bile ducts, a cholestatic injury, like a gallstone, we would expect to see the ALP and GGT spike significantly.
So we have confirmed the liver cells are actively rupturing and leaking workers into the bloodstream. Next, we look at the ratio between ALT and AST. In this patient, the ALT is higher than the AST. When we combine that ratio with the clinical context, a 45-year-old patient with fatigue and a BMI of 34 non-alcoholic fatty liver disease, or NAFL, rises right to the top of the differential diagnosis.
Because of the BMI.
Yes. The excess fat accumulation in the liver creates oxidative stress, slowly damaging the cells and leaking the enzymes.
But the elevated ALT doesn't confirm AFLD definitively, right?
Yeah.
You still have to rule out other suspects.
Oh, absolutely. ALT is the flare gun that tells us where to look, but it doesn't always tell us who fired it.
Right.
I must rule out competing causes of hepatocellular injury. I need to know their alcohol intake because alcoholic liver disease typically presents with the reverse ratio and AST that is higher than the ALT.
Oh.
I need a full medication history. Are they taking statins or herbal supplements that could cause drug-induced injury? We also need to run viral serology to ensure this isn't an underlying hepatitis B or C infection causing that fatigue.
Right, you can't just stop at the first assumption. Well, this case perfectly illustrates the mission of our deep dive today. A single data point on a screen is just the opening sentence of a much longer clinical conversation.
It really is.
Let's distill the key takeaways from today's journey. First, ALT is an intracellular metabolic enzyme. Its presence in your blood is a marker of cellular injury, not an overall measure of liver function.
And second, That number is the product of a staggering workflow.
Right.
I mean, it relies on avoiding pre-analytical errors, utilizing IFCC standardized assays that measure invisible light absorbance, adhering to strict statistical quality control.
The Westgard rules.
The Westgard rules, yes. And depending on the vital expertise of biomedical scientists to validate the reality of the chemistry.
So the next time you get a routine blood test, don't just scroll past the acronyms. Recognize that a dedicated team of unseen scientists, rigorous laboratory infrastructure, and incredibly precise ultraviolet physics were deployed just to give you that single validated data point.
It entirely reframes what it means to be truly well-informed about our personal health metrics.
It does, and it leads to a final thought I want to leave you with. We are accelerating rapidly into an era of at-home health testing and wearable tech.
Oh, for sure.
Silicon Valley envisions a future where we check our complex biomarkers as easily as we check our text messages. But if the absolute gold standard of laboratory medicine requires multi-million-dollar analyzers, rigorous statistical rules, and intense human scientific judgment just to prevent a shaken plastic tube from causing a false diagnosis,
Yeah.
how much absolute faith should we really be putting in the instant automated health metrics we carry in our pocket?
That is a phenomenal question to consider as technology evolves.
Definitely something to mull over the next time your smartphone tells you that you are perfectly healthy or warns you that you are sick. That's all for today's deep dive. We'll see you next time.